Tiff, how are you?
Yeah. How are you, Sam?
I'm good,
Happy
thank you.
New Year. Am I still allowed to say that?
Yeah, just about. It's
We're
still
still in
January.
January.
No? What is it, 21st? 20th?
24th.
Is it? Oh, God. Oh, God. Oops.
Well,
24th of Jan.
yeah, crazy. Where did the time go? But yeah, welcome back to Breakthrough, the podcast with Sam and Tiff. We're excited to go over some of the breakthroughs from 2025 in the world of medicine. And I'm Tiffany, your co-host. I'm an entrepreneur, scientist. I co-founded Gambit Bio, where we're doing saliva-based testing for early cancer detection. And I'm also part of Infinitope's, a cancer vaccines company.
Amazing. And maybe we'll talk about those guys a bit later as well. I'm Sam. I'm a medical doctor. So should we talk about... So we're going to talk about what our opinion on the biggest medical breakthroughs from 2025.
Yeah.
Right?
There are lots. Sam wrote an amazing Substack piece. Check it out on the top 10. But today we've decided to focus on our favorite 2. So 4 total.
Yes.
2 from you, 2 from me. But yeah, I thought, let's kickstart with talking about your favorite GLP-1s.
I've grouped it as metabolic.
Yeah, So under the group of metabolic medicine.
Yeah. My top 10 list from last year, I cheated a little bit. It's more than 10, really.
When I started reading, I was like, Sam, this is more than 10.
More than 10. I cheated a little bit. It's 10 themes. It's 10 themes, and within each theme, there's multiple kind of breakthroughs within each one. But the thing is, if you don't do it like that, then you leave too many things out, and that's what I don't like.
I liked it though. So comprehensive.
It is long.
Highly recommend it for anyone who wants a great comprehensive overview of breakthroughs in 2025 in medicine. But yeah, so, everyone talked about GLP-1s throughout the past few years, but what actually changed in 2025?
Good question. So the reason this is so important is because by the year 2050, the projection is that more than 4 billion people in the world will have overweight
half the
or obesity.
planet.
Exactly. So it's over 50%, 4 billion people. That's a staggering number. The numbers are just getting bigger and bigger. And so that's why it's so important. And last year, we saw a number of new modes of action come through in the metabolic and obesity space. New phase 3s, firstly. So there's a drug called CagriSema, which is an interesting one because it's a GLP-1 and an amylin receptor agonist. Actually, it's 2 separate molecules combined into one drug. And that came out with new data. There was a drug called mazdutide, which is once a month. So typically, GLP-1s are once a week. This one is once a month. It's also got a glucagon receptor agonist on it, so that's a new mechanism. Retatrutide is where, let's be honest,
I'm impressed you can pronounce all these things.
Retatrutide is causing a huge amount of excitement right now in the obesity space. It's a triple agonist. Which I find fascinating on its own, because how many drugs are there that are triple agonists that you can think of?
Yeah, I don't think I can think of one.
No, exactly. This is and the fact that it's a triple agonist, super unique mechanism, and so in the phase 3 trial when it came out, it produces almost 30% weight loss in less than a year.
Wow, that's crazy. And why are people so excited about, for example, the new mechanism of action through amylin? How is it different to GLP-1s?
Yes, and I think you were early on this one. And there is another one as well. It's a pure amylin receptor agonist called aloralintide, which had a positive trial last year. So not only is it combined with GLP-1s, but also a pure amylin agonist on its own. And the reason that people are so excited, I think, is because it causes you to feel full rather than stopping you feeling hungry. It causes you to feel full.
Because GLP-1s stop you from feeling hungry, but then amylin analogs help you feel full and satiated.
Exactly.
Yeah.
And I think it's interesting because at the moment, lots of regular people in the world know about GLP-1s, right? No one knows about amylin receptor agonists, but there's definitely a theory that amylins will come and overtake the GLP-1s.
Especially with the patent cliffs coming out.
Yeah, exactly.
Wow.
So the future of obesity is definitely in amylin.
Nice.
Well, there's a lot of people think that the future of obesity is in amylin rather than GLP-1.
Yeah, and those are obviously treatments. But then in your Substack, you also talked about preventive therapies, especially in the cardiovascular space.
Yes.
So what excites you as a doctor in preventive medicine?
100%. So there were new phase 1 trials for one and done cholesterol therapies. So one is from an Eli Lilly company called Verve Therapeutics. They have a gene editor. Edits a gene called PCSK9.
Yeah, we covered that, didn't we, in one of the deals?
And it's a one and done therapy. So it goes to the liver, edits PCSK9, switches it off, and lowers cholesterol. And in theory, you just take it once, and cholesterol is low forever.
That's amazing.
So there was new
like
data
curative, basically.
Exactly.
Yeah.
That was super exciting. There was another new medication last year which had a positive phase 3 trial, which lowers triglycerides.
Ooh.
So at the moment, I am working in surgery. You see a lot of pancreatitis. Most pancreatitis is caused by gallstones, but a significant proportion is caused by high triglycerides. At the moment, if you've got high triglycerides, very high risk of pancreatitis, you can't do anything about it.
Yeah.
This is a drug that lowers triglycerides, and last year's breakthrough lowers the risk of pancreatitis by like 85%.
Wow. So it's like 2 birds, 1 stone.
Exactly. And that's the very first medication ever shown to lower the risk of pancreatitis in like in the population.
Wow.
Very, very big.
How does it work?
It is a antisense oligonucleotide.
Wow.
It lowers prevents expression of a gene called APOC3, which is a triglyceride regulator.
Wow.
And so it drops triglycerides and drops pancreatitis too. Super cool.
Exciting.
Yeah.
Oh my gosh. I see why like that's the first one on your list of 10 on your article.
Metabolic.
Yeah, metabolic is huge. And it's probably the biggest market right now.
Yeah.
As well.
Exactly.
It's bigger than cancer, I would say.
Possibly, yeah. The other one is MASH. So MASH is this chronic liver disease.
Which used to be called NASH.
Exactly, NASH.
But now it's MASH.
Non-alcoholic fatty liver disease, now called metabolic dysfunction-associated fatty liver disease. And only in 2024 was the first therapy for MASH. And now, if you look at the pipeline of like emerging therapies, it's extremely hot. And last year, another therapy was approved for MASH, which was semaglutide.
So everything is starting to get like pushed out from the pipeline.
Yeah, exactly. It's one of the hottest areas in biotech for sure, is MASH.
Wow.
Yeah, so very exciting. And for good reason too, because MASH can cause liver complications, can cause liver cancer, can cause cirrhosis, can cause liver failure.
Yeah, all the other comorbidities that go along with it, basically.
Yeah.
Yeah, huge market.
100%.
Wow. Cool.
So
So that's the first one. That's your first fave, the metabolic medicine breakthroughs.
Yeah, exactly.
I'll switch gears a little bit. So one of my favorites was, and we talked about this previously as well, in the world of gene editing. So yeah, gene editing has been in the public imagination for a while now, especially with CRISPR from a few years ago. People think about like designer babies and designer genomes, etc. But I would say 2025 was the year where it, went from being a futuristic thing to actually being clinical. And obviously I have to give a nod to Vertex, since they came out with the first widely approved CRISPR therapy for sickle cell disease and beta thalassemia.
Amazing.
But the Vertex one, I would say, proved that you can scale, because they can manufacture at scale, this ex vivo gene editing tool where you have to take the patient's cells, edit it, and put them back. It's ridiculously expensive, but it's possible, and it could be curative. But what was shown in 2025 was that this can be done at speed with precision for just one patient, which was groundbreaking. And I know you love this story as well, but it's the story of baby KJ, as he's known in the public. But this really defines the new era of gene editing, because baby KJ has a very ultra rare, also metabolic disease called, I'm going to say this right, carbamyl phosphate synthetase 1 deficiency, CPS1 deficiency. And this basically means without treatment, toxic ammonia builds up in the body and specifically in the blood and can cause brain damage. And usually the patient would not go past early childhood and there would usually be death. So it's kind of like a death sentence, this diagnosis. But instead what happened here was extraordinary because, this team of scientists, they firstly made a genetic diagnosis. So they found out exactly where in the gene the mutation happened because it can happen in different spots and you need, a different therapy for different mutations in that gene. Then they made a patient-derived cell line. So they took baby KJ cells and then they expanded it so then they can test the different gene editing tools in the lab and optimize it before they gave it back to baby KJ. And then finally, they designed something called a base editor to correct this, genetic mutation. And the most impressive thing is they did this all within the span of 7 months, which is just record breaking.
So wild, right?
Right?
I mean, so he hadn't even been born, and then 8 months later, they'd developed a brand new therapy and dosed him. I mean, it's incredible.
Yeah, like in the world of medicine, 7 months is like nanoseconds.
Yeah,
when things are usually
100%.
measured in decades.
Exactly, yeah.
Yeah, and now the baby's like more than a year old, and he's walking and healthy and developing.
There's this amazing photo of him at Christmas, like walking around the Christmas tree, which is incredible.
It's just like the most fulfilling, sweetest image. Yeah, obviously for the family, but also like for scientists and the world of medicine is so inspiring. But yeah, like just to zoom out of like this upgraded tool, for the audience, everyone's obviously heard of CRISPR, and you can think of this as like a pair of scissors that chops up both strands of your DNA. But the bad thing is it might cause unwanted mutations and errors, which might have downstream, unwanted symptoms or effects. But here, these are like the new versions of gene editing tools, and that includes base editing and prime editing. And here, base editing was used. So if you think of your genome like a storybook with words and sentences, then the base editor is only changing one letter. So you can only substitute one letter at a time. Whereas I would describe the prime editor as kind of like something like a control, find, and replace tool where you can correct like insertions and deletions and like change words and sentences at the same time. And both, importantly, only edit one strand of the DNA. So you don't edit two strands like CRISPR, so you have fewer side effects potentially. But yeah, Sam, you also mentioned like, these genetic editing tools sound too good to be true. And people are like, what are the long-term effects?
Yes.
So do you know if there are any
exactly.
long-term
and
data?
There's
like
always
that?
the criticism, right? Yeah, but what does it work long-term? Is it safe long-term? And so I would also highlight, so it's a different approach. So it's not a direct gene editing approach, but it is
it's one form
gene
of
therapy
gene editing.
approach.
Yeah.
For two conditions where there's very, very long-term data now. One is called, the condition used to be called bubble boy disease because it usually affects boys and they used to have to go and be kept in bubbles to avoid
To not interact with the outside world.
Exactly, because they're so susceptible to infection. Yeah. So the medical name is severe combined immunodeficiency. So you're deeply immunosuppressed. And so you have to be protected.
So you cannot risk any infections.
Exactly.
Wow.
But there were some gene therapies that happened 15 or so years ago, and the follow-ups show that that long-term cure is possible. So it's over the course of 15 years, which is a long time course.
Yeah.
It's safe and it's effective.
That's
which
amazing.
Is perfect.
So usually, and all these, patients, they're living and, otherwise without the therapy, like the death rate would have been, like none of them would have been alive, right?
Yes, exactly.
That's incredible. And that, goes to show how promising these gene therapies or gene editing tools are. So, fingers crossed for the long-term data for these CRISPR and base editing tools. That's awesome.
100%.
Yeah.
You mentioned, just quickly on prime editing, you mentioned prime editing. That was done in humans last year for the first time too.
Wow.
So in a condition
Do you know
called
which
chronic
disease?
granulomatous disease.
gosh,
So
what
it's
is
also a
that?
rare genetic disease.
Yeah.
And prime editing was used in human patients for the very first time, and it looks like it's effective. So we only have data
Fingers
from the
crossed.
first few months, but it looks like it's effective so far.
That's
so pretty amazing.
incredible.
There's a company as well called Prime. What's it called? Prime Medicines, I think. They are focused purely on prime editing, as you'd expect.
As the name suggests.
And that's the biotech that's building these prime therapies. So it's pretty cool.
Yeah, that's so cool. Rewriting genes with precision. That's where we're at right now in personalized medicine. Super cool.
Exactly.
Well, I think moving on to your next favorite breakthroughs, breakthrough from last year. My question is, can we train the immune system with the same amount of precision as well? So moving on to
good question.
transplants.
When I did med school, my favorite part of med school, the whole thing, was transplant medicine. And I was in the Churchill Hospital in Oxford. I think it was a Churchill. Yeah. And they do transplants, and it's incredible. They do kidneys, they do pancreas, they do uterus. It's crazy.
Really? Wow.
They do. They even do like small bowel. It's amazing. I love transplant medicine. They don't do livers, actually. I think they might do now, but they didn't when I was there.
That
And they
must be
certainly don't
incredible.
do
You see
this-
just two people on the operating theater. And
Not at the same time.
okay. Oh, no, you get
Yes.
it from one person,
Yes.
and then you immediately
Yes, exactly.
go to the
So
patient
usually
receiving
they
it.
put it on like ice, or indeed they like perfuse it. They put it in a machine to like perfuse it with blood to
Yeah.
keep it going. That's actually a recent breakthrough, by the way.
Yeah.
That machine.
Another one.
But yeah, so it's amazing. So I love transplant. Oh,
Yeah.
and the other thing they do is like islets.
Oh, right. Which we also talked about.
Exactly.
Yeah.
So one of the transplant breakthroughs from last year concerns type 1 diabetes. So islet transplants is commonly done to replace the beta cells. They produce insulin. They're lost in type 1 diabetes. The issue with how we do it currently with donor-derived islets is supply, right? How are you going to get all these islets? Where are you going to get them all from?
Yeah.
So if you could produce them via a stem cell line, so you produce stem cells, derive them into islets, and then transplant them.
Yeah.
That was done last year for the first time in a series of human patients successfully with type 1
Amazing.
diabetes. Super cool. Then the next question is what you'd say is, okay, but if you have to take immunosuppression for life,
Yeah.
which you do with that approach, then it's not a cure, right?
Yeah.
So the other one that happened last year, another breakthrough, published in the New England Journal, they took islets, they edited them to prevent them stimulating the immune system. They did gene editing, like we were just talking about.
Yeah.
And then they transplanted them into a human. And without any immunosuppression at all, they were able to produce insulin in this type 1 diabetic who'd been diabetic for like decades. For
That
the
was
first
an
time, he
incredible
produced
study.
insulin. Amazing.
It's because they were able to edit the cells in such a way that it doesn't induce the donor rejection or donee. What's the name?
The acceptor? What do you call it?
I have no idea. Oh God, English is my third language. But yeah, you know what I mean. Like
Yeah.
it's not rejected
Yes.
by the person receiving it. It's so smart because we know the mechanism and they're able to suppress those specific mechanisms.
Yeah, exactly. I think
Yeah.
it was they suppressed the macrophage-induced immunity and they suppressed T-cell-induced immunity.
Yes, through the MHC complex.
You're the expert on
There
that
you
one.
go.
We'll talk about
That's
that later.
Incredible. So what happened this year in terms of transplants? Because you also mentioned like pig organs,
Yes.
right? What happened there?
So there
It
was-
seems like sci-fi.
I know, right? It's so there was 3 firsts in pig organ transplant. So the big issue in transplant is just supply. So if transplant surgeons could have like even like a few extra organs to transplant every year, they would bite your hand off for it, right? They would take it 100%. So the issue is supply. So one animal we could use organs from is the pig.
Pigs.
And so this year there were 3 firsts in pig-derived transplants. So the first report of kidney transplant from pig to a human, gene edited, very cool. The first liver transplant, so it's much more difficult to do a liver than a kidney.
Yeah.
the first liver transplant from a pig to a brain dead human. So it was an experimental study.
Why is it harder?
It is much more like anatomically challenging than a kidney. It's I don't really know. It's much harder.
I don't know. I'm just
it's much harder.
Yeah, somehow like kidneys. Yeah, because you hear about it all the time. Like kidneys being taken and transplanted, but not livers.
Yeah, exactly.
So I was like, why?
Liver is harder.
Yeah.
The precise reason, I don't know.
We'll find out. I'll Google this afterwards.
Yeah.
Yeah.
The other one was lung. So the first ever lung transplant from a pig to a human as well, which, for the first time ever, that's amazing.
That's crazy. I mean, so many people have collapsed lungs. Like they're walking around with like one lung functioning. So this would, make a huge difference.
Yeah. Exactly. Transplant I love transplant, and last year was a very, very good year for lots of firsts in transplant medicine.
Incredible. I can't wait. I mean, it is a supply and demand problem. So, wow.
Exactly. What do we got next, Tiff?
Well, I'm going to try not be biased, but it's hard not to be. So next up is, I think our favorites, both our favorites, cancer vaccines. And before I start, full disclosure, I am obviously an early employee and scientist at Infinitope's Oxford Uni spin out.
But it's the
making
future.
Cancer vaccines.
Cancer vaccines are the future,
It
so
is
we can talk
future.
about the future.
Yeah, it really is. Precision medicine could be used as prevention, but basically early stage intervention as well.
Yeah.
And what was exciting about cancer vaccines in 2025 was not an overnight miracle, but rather it's another stack of evidence through clinical trials, late stage, phase 2 and phase 3, that are really showing that cancer vaccines are effective and they're saving lives in really difficult to treat cancer types. And, as a background, for those who don't really know about cancer vaccines have had quite a checkered history because, the idea works beautifully on paper, train an immune system to identify the cancer cells and kill them with cytotoxic T cells. But the reason why cancer vaccines haven't worked until recently is because the cancer is really good at doing its job, hiding and escaping from the immune system. And the way to hunt these cancer cells down is to find these things called neoantigens. They're like little flags that the cancer cells have to say hey, look, I'm a cancer cell. And I'm not a normal cell, but they're really good at hiding away from the police of your body, the immune system. So a way to produce better vaccines is to find those little flags that the cancer cells are kind of waving, but in a not very obvious way because they don't want to be found. And just a quick note, even though when we call these cancer vaccines, but unlike the vaccines that people think of when they hear COVID vaccine or the flu jab, this is not a preventative therapy. It's usually therapeutic. But the logic is the same, where you train the immune system with bits of the weakened virus, or in this case, bits of the cancer neoantigens, which is basically fragments of proteins, and then you get the police force in your body, the immune system, the white blood cells to hunt these these CD8 cells, and they will hunt down your cancer cells and kill them. So every cancer has these mutations, and they get turned into these fragments, and then you hopefully find these fragments. And now we can do it better than before because of sequencing technologies for the DNA, RNA, and for these peptide bits through immunopeptidomics and mass spec technologies. This year there were particularly 2 trials that we will pay attention to. The first one is in kidney cancer. So in this study, kidney cancer patients had their tumors removed, sequenced, and then vaccines were made personalized to each patient because each patient might have different tumor mutations. And then the results were striking because historically 50% of these patients would be expected to relapse. But in this case, in the vaccinated group, over, I think it was a 3-year follow-up, none of them had a recurrence, which is mind blowing. Right, it's still going on, but ongoing, but that's really promising.
Highly encouraging.
Exactly.
Signal, yeah.
And the second one, I don't know if you want to talk about it, Sam, but it's in pancreatic cancer, which is the prognosis for pancreatic cancer. It's brutal, right? It's 3% survival after the best standard of care for 5 years.
Disease.
Yeah, exactly.
Which hasn't, by the way, if you look back historically at the last 20, 30, 40 years of metastatic pancreatic cancer, there's been zero progress. It's been 3% for forever.
Yeah,
so we desperately need new. So this one was for resectable disease, I think.
Yeah,
but still, we desperately need new therapies for pancreatic cancer.
Exactly. This is like a really great first step.
Yeah.
And obviously, when you prove a new therapy, it's best to do it in a low hanging fruit population. So doing it in resectable disease gives you another chance to get the funding to do it in the metastatic disease. But yeah, in this case, like in the personalized vaccine trial for pancreatic cancer, patients who mounted a really strong vaccine-specific T cell response, showing that the T cells were going for targets that the vaccine had, the outcomes were dramatically better, and with many being cancer-free, like years after the vaccine. And this doesn't normally happen in pancreatic cancer. So
no. Super cool.
Yeah.
There was another one last year in pancreatic cancer, specific. Remember we talked about it, specific for RAS.
Yes.
So most more than 90% of pancreatic cancers are driven by RAS mutations.
The oncogene.
Exactly.
Yeah.
So they built a vaccine specific for the RAS mutations in order to, again, try to prevent recurrence of pancreatic cancer. So it's super cool. The other vaccine one, I follow all of the vaccine trials, by the way. At least I try to. I try to actually build like a table of the cancer vaccine trials. It's difficult to do because it feels to me it feels a bit all over the place.
There's so many different types. It's like
kind of difficult to
cancer types. Some of them are multi-cancer.
Yeah.
Delivery system. Is it DNA, RNA,
it hasn't quite,
peptide?
Formed into a nice kind of categorized field yet. I don't think.
Yeah.
That's because it's new though.
Exactly.
But the first big cancer vaccine trial was in melanoma by Moderna.
Yeah.
And what bothered me slightly was that if you look at the p-value for control versus vaccine, it's not quite significant for looking at event-free survival, progression-free survival. But and that was at 3 years, I think. But just recently, they had 5-year data come out, same trial, but 5-year data. And now
it was positive and statistically significant.
Exactly. And so I looked at it and I thought, phew. So it's very nice. It's in melanoma. It's pembrolizumab, anti-PD-1 versus pembrolizumab plus vaccine, personalized. Yeah. And so the vaccine group had statistically significantly longer progression-free survival versus the other group.
Yeah, I think like when I looked at like the last phase, the studies show half the chance of recurrence.
Oh yeah, big effect as well.
And now
50%.
Is it still 50%?
It is.
That's amazing.
So it's a massive effect. It's not, and we know, by the way, that pembrolizumab is very, very, very effective on its own.
Yeah, especially in like the high tumor mutational burden tumors.
And that's what's so exciting, I think, about cancer vaccines too, is because we know they're not going to have so many side effects. We know the side effects of vaccines. You feel a bit ill, maybe you got a bit fluey, you got a bit of pain for a couple of days, and then that's it.
Yeah.
That's it. There's no long lasting.
It's not like chemo. It's not like some of the checkpoints which could have, very bad side effects. So you could
add it on top of these more toxic therapies to boost their effect without causing
negligible side effects.
That's why I think one of the reasons it's so exciting.
Yeah, me too. That's why it's like I feel like it is the future of oncology.
It is 100%.
Yeah, it's like a really big upside, and like it will boost the effects of the existing therapies, especially checkpoints, or really I would say it gives hope to cancer types where checkpoints don't work very well.
Any in mind?
Yeah. That could be a whole episode on its own.
Next time. Okay.
Next time we can talk about...
We reached the limit. We should say the limit is 30 minutes.
Yeah. We're trying to... 2026 resolution for Sam and Tiff is to keep everything under 30 minutes.
So if you feel like we were fast, that's
let
fine.
us know. Well, we will try. We're trying to appease to both scientists and like the general audience who want to learn more about science.
Yeah,
Yeah.
exactly.
But yeah, come join us next time where we will talk about what we're looking forward to seeing in 2026 in terms of science
predictions.
and
2026
deals.
predictions.
Yeah, 2026 predictions.
And one of them, this is clickbait. One of them is going to change the
cliffhanger.
world. It's going to change the world.
Yeah.
So there we go.
I think I know what you're talking about. There are a few. See you next time.